Hi everyone, I'm Dr. Erica Shelley from Best for Bees, located in Ontario, Canada, and I'm here to talk to you about the coolest thing you've probably never heard of, Entomovectoring. Yes, bees are about to be your new delivery drivers, but don't worry, they don't demand tips or miss your packages. Like many of you, I'm sure each morning I wake up and I'm invigorated because the work I do has purpose. Now, a bee talk wouldn't be a bee talk if I didn't tell you about the importance of bees and why they are struggling. Bees are an important component of our food network. However, throughout the world, and I have traveled to know, up to 50% of beehives die each year. And that is a significant threat to our food security. For 15 years, Best for Bees has been working to solve problems with bees and bee health. And in 2020, we were recruited to work on a project with Dr. Peter Kevin at the University of Guelph. Now, I need to introduce you to the one and only Dr. Peter Kevin, a true legend in the world of bee science. Dr. Kevin is like the David Attenborough of bees, except his documentaries don't air on TV. They're published in over 350 scientific journals. He's worked on everything from studying Arctic plants to understanding bee vision, and he's even had a bee named in his honor in Chile. That's right. He is also the guy who made it possible for bees to deliver more than just honey. Dr. Kevin's groundbreaking work on entomovectoring using bees to deliver beneficial agents laid the foundation for what we're doing today at Best for Bees. And when you've got a bee and a plant expert like him pioneering a project, you know it's going to have a big impact. In 2020, we were thrilled to join forces with him on the Entomovectoring project funded by one of Canada's largest grocery chains. It's been a partnership that brings together academia and industry to tackle one of the most pressing challenges in beekeeping, improving hive health. He's had a career spanning decades, and his work is the reason we're able to explore new ways to enhance bee health using natural, sustainable methods. So if you take anything away from today, it's that bees, with the help of Dr. Kevin, are not just pollinating, they're also helping us innovate. Okay, so here's where things get really fun. We're going to talk about bee vectoring. Now, I'll admit just a few years ago, I didn't even know what entomovectoring meant, and let me tell you, spell check still doesn't like it. But it's a simple concept. Basically, we're using bees, the ultimate multitaskers, to deliver more than just pollen. Here's how it works. We add a powder to this tray up here on the far left, containing some beneficial agent. And that, as they exit, the bees walk through a powder. Originally, we used bumblebees, but honeybees can also be used. As the bees leave the hive, they walk through the powder and it sticks to them. Then, as they go about their day pollinating flowers, they're also delivering these beneficial agents, kind of like tiny delivery trucks, but a lot cuter and far more eco-friendly. The real beauty of this system is in its precision and sustainability. Instead of spraying chemicals all over crops, the bees target exactly where the treatment is needed, and they do it over an extended period. No need for reapplication or waste. Plus, they're out there every single day, covering flowers over a longer period than a one-time chemical spray could ever manage. What's even better is that this method is perfectly in sync with the bees' natural behavior. They're already out there pollinating, so why not let them drop off a little something extra while they're at it? Now, we usually use something called a biological control or a biocontrol in the powder. It really could be anything, but that historically has been what's been used. And there are primarily two types of fungi that have been used, either an entomopathogenic fungi or a necrotrophic mycoparasitic fungi. The first, the entomopathogenic fungi, like Beauveria bassiana, attacks and kills insects. This fungus is naturally found in the environment and it works through multiple methods, both breaking down pests physically through mechanical action and chemically using enzymes that will actually penetrate through their shells. The second type, the necrotrophic mycoparasitic fungi, targets other fungi I like to kill them, eat them up, and we get the good fungi taking over the bad fungi, like black mold in strawberries. So these fungi, they help control diseases like mold or fungal infections in plants. These fungi are fantastic because they're long lasting, reducing resistance in pests and diseases. Many of you are too familiar probably with the challenges of varroa mite treatments. You have to remove honey supers to apply the treatments, and it has to be done within very specific temperature ranges. That's where Dr. Kevin's vision came into play. He proposed a system where the bees themselves could distribute treatments throughout the hive without disruption. This concept, which we call inspensing, allows bees to walk through a powder at the entrance and naturally spread it throughout the hive. No need to lift heavy honey supers or worry about temperature limitations. Inspensing works seamlessly under various conditions, providing continuous treatment as bees enter the hive. And let's be honest, traditional mite treatments are actually no walk in the park. First, you have to haul off those honey supers that weigh up to 100 pounds. Then wait for perfect weather and hope you don't accidentally knock off the queen while you're at it. It's like the beekeeper Olympics, and no one wants to compete every time mites show up. Inspensing, on the other hand, is a breath of fresh air. Bees just walk through a powder at the hive entrance, and that's it. No more heavy lifting, no need to time treatments around the weather or temperature. This works rain or shine, day or night. Plus, the bees distribute the treatment everywhere inside the hive, making it more efficient and less invasive. And for the hobbyist beekeeper who might be a little nervous about opening up their hive, this is the dream. There's no need to dig in, risk upsetting the hive, killing the queen, or worrying about getting stung. Just sit back, relax, and let the bees do all the heavy lifting for you. So, how did Best for Bees actually end up working with Dr. Kevin on this innovative inspensing technology? So, for the last several years, we've been providing a wide range of bee related consultation services to companies. Want more seeds produced? Best for Bees will do traffic study on bumblebees or honeybees. Concerned that your product might be harmful to bees? We're the third party assessment team you call. We even taught bumblebees to use touchscreens to test their memory. Yes, touchscreens for bees. That's how far we go to understand pollinators. We've also been working on more traditional projects like setting up corporate beekeeping programs under our business bee service, long before companies like Best Bees and Aviol made it a popular business model. As for myself, I've got a PhD in molecular and medical genetics, and during grad school, I worked with fungi to study DNA repair. Back then, I never thought my two worlds, fungi and bees, would collide until Dr. Kevin reached out to us at Best for Bees for help with that inspensing project. That's when Best for Bees became part of this groundbreaking work. Now, what you see in front of you here, that is the Protectabee. That was the answer to getting powders inside the hive. So let's talk about how we came up with the Protectabee. We actually borrowed some ideas from an old piece of beekeeping equipment, the Bee Escape. A bee escape uses cones to gently move bees out of the supers. When you look at those cones, the bees do not like to go in those teeny tiny ends, and we're going to talk a little bit more about the red color and why that's there, but they don't mind going in the larger part, and as it narrows, their little heads poke right out. In the evening, when it's cooler and you're using a bee escape, the bees actually head down to the babies in the brood chamber away from the honey. They go through the cones and they can't go up. Voila! So this allows the beekeeper to come back the next day to remove the honey boxes without any bees inside. We took this clever concept and added a twist. For the Protectabee, we designed an entry system with two pathways, one for bees going in and one for bees going out. And here's the fun part. We incorporated the color red into the design. Why red? Well, bees don't have a red receptor, so to them, red mostly looks like black. Is it invisible? Not exactly, but it does appear as a tough to decipher black blending in with its surroundings. Now, don't worry, the bees are much smarter than they look. They navigate through the protective bee using signals, scents, and orientation flights to figure out their way through the system like a seasoned traveler at an airport. It's bee traffic control at its finest. Let's dive into how our star product, Protect-A-Bee, actually functions inside for bee vectoring. The system is pretty straightforward, but highly effective. At the entrance of the hive, bees walk through a powder filled in drawer, which contains whatever treatment or biological control agent you want to introduce to the hive. Now, without the vectoring block in place, bees might just walk along the ceiling and avoid the powder entirely. But once the block is installed, bees have no choice but to walk through the powder, ensuring it sticks to them. As the bees move around inside the hive, they naturally distribute the treatment, covering every nook and cranny, so it's delivered precisely where it's needed. This system is designed to use the bees' natural movement patterns, minimizing beekeeper intervention. No need to constantly open the hive or disrupt their natural flow. Plus, it reduces the amount of product needed since the bees themselves do all the distribution work, making it more efficient. All right, so here's the fun part. I have a video that shows the Protective Bee, how it really works. You'll see our star bee flying to the hive like a pro, entering through the end cone and getting a nice dusting of powder as she walks through. Watch closely as it goes about its business inside the hive, spreading the powder on the frames. This video gives you a sneak peek at how effortlessly our bees handle the inspensing process. Just last month, our research paper on the Protective Bee was published, showcasing some of our findings. I'll highlight a few key points today, but for those of you who are interested in all the nerdy details, you can check it out in Frontiers in Bee Science. This paper delves into the engineering behind Protective Bee and shares the studies proving how effectively bees can vector powders into the hive. It's bee science at its finest. So we've done the science, cue the lab coats and microscopes, and here's what we tested. First up, a fluorescent powder. Nothing says serious research like glowing powder under a black light. We divided the hive into zones. We've sampled eggs, larvae, honey, pollen, and even bees and mites, and tracked how well the bees spread the powder throughout the hive after 24 hours. Next, we tested oxytetracycline, a common antibiotic in beekeeping. The bees vectored it for us while we measured bacteria levels using microbial inhibition tests to see how much bacteria was wiped out. And for the grand finale, we used Beauveria bassiana, a biocontrol fungus. To check its distribution, we used quantitative PCR to detect the presence of the fungus on larvae and mites, making sure the bees spread it effectively. The results? Well, first of all, the fluorescent powder was found in almost 80% of samples, showing a solid distribution actually throughout the entire hive. As for the antibiotic, it was spread evenly throughout the hive without ever opening it, proving the bees handled delivery better than any postman. We're going to come back to those results. But the real star for us when we were checking for mite treatments was Beauveria bassiana. Using quantitative PCR, we confirmed it in 86% of larvae and a whopping 92% of varroa mites. The bees didn't just deliver, they nailed it, making Protective Bee a potential game changer for in-hive pest control. So, what's next? Well, we've built a bee vectoring system for inspensing that works wonders at delivering treatments into the hive, removing the usual hurdles like heavy lifting, bad weather, and beekeeping nerves. While we were working on our project, Dr. Shepard's team at Washington State University published a study on a new strain of Metarhizium that thrives in the hot, humid conditions of a beehive, much better than our current Beauveria bassiana strain. It's a prime candidate for varroa mite management, and it opened our eyes to a whole new set of possibilities. Right now, there are a number of potential products we could use: RNA interference, amino acids, biocontrols such as those fungi we mentioned, essential oils, you name it. If it can be powdered or sponged, we can get bees to vector it into the hive. Of course, there's a small catch: approvals. As I'm sure many of you know, getting regulatory approval is like watching honey drip. It takes time. But we're also working on research with pollen in an outgoing tray to boost crop yields. Bee vectoring to crops? We haven't jumped into that pool yet, but it's definitely on the horizon. So for now, we collaborate, we innovate, and we wait for those all important approvals. So here's something exciting though. While we were using oxytetracycline, that antibiotic, as a way to monitor powder distribution, we actually uncovered a new method to add antibiotics to the hive without needing to open it. And here's the best part: antibiotics might not require the same long approval process as other products. By delivering antibiotics without opening the hive, we're not only reducing disruption to the bees, but also preventing the spread of disease within infected colonies. This method could be a game changer for managing diseases while keeping the hive safe and sound. So, antibiotics in the hive is used typically for two different types of bacteria: American foul brood and European foul brood. American foul brood, it is nasty, awful. It's going to kill your hive. And generally, if you have an American foul brood hive, it is recommended to burn it. So, we're not using antibiotics in AFB hives to deal with an active infection, but there are some places, such as where I live in Ontario, that prophylactic treatment with antibiotics in the spring will help to stave off future infections. You can also treat other hives in an apiary that has been affected with American foul brood. European foul brood is a different bacteria altogether. It doesn't form spores, so it doesn't tend to stay in equipment the same way that American foul brood does. American foul brood can actually stay the spores in equipment for 50 years or more. But the European foul brood does arrive in early spring when there is cool, cold weather. And you can actually use antibiotics during an active infection. But in North America, that is only with a veterinary prescription. You're also going to want to treat your other hives in those affected air apiaries. While using antibiotics inside a hive may not be a popular choice, it can sometimes be the necessary treatment to contain these devastating diseases. So, after seeing the antibiotic distribution looking good in our research, we decided to compare it with the conventional method. Usually, antibiotics get mixed with powdered sugar and placed on top of brood frames. But here's the kicker: it was a super rainy summer when we were doing our experiments, and getting into the hives for the conventional tests was a huge hassle. With bee factoring, we had no such issues. It was smooth sailing, and we easily got our results. Bee factoring delivered antibiotics in 80% of the samples, and the conventional method hit 90%. Statistically, not a difference, and you might even say, oh, there was more with the conventional method, but we're going to show you in a later slide how this actually was a miss with a lot of those samples. Bee factoring can be just as effective, or actually more effective, than conventional methods, and it's much easier when the weather isn't on your side. Now let's talk about antibiotic resistance and some of the key issues contributing to it. So first of all, improper dosage and duration. When antibiotics are given at incorrect dosage or for the wrong amount of time, we're either not killing the bacteria or worse, creating conditions where only the resistant ones survive and thrive. We also have antibiotic resistance that can rise from prophylactic use. When people are overusing antibiotics as preventative measures, that can lead to selective pressure, favoring bacteria that are naturally resistant, which can actually lead to long-term resistance issues. And finally, lack of veterinarian oversight can be a big issue. In some places, when we see inconsistent use, we find that's a fast track to resistance. Veterinarians, they know how to mix it correctly. They give you the correct way to use it for your area, depending on what you're using it for. So what does this all mean? Improper dosages, prophylactic use, and lack of veterinary oversight are key drivers of antibiotic resistance. By using antibiotics correctly with the right dosage and duration under proper supervision, we can reduce the risk of bacteria developing resistance in our hives. This is where responsible antibiotic use becomes crucial. And now let's see how bee vectoring can help. We're going to talk about accuracy. Now, this is a box plot just showing the distribution of oxytetracycline levels. Now, you might not be familiar with box plots, and let me just describe to you what these X's mean. These X's represent the median, the midpoint of our data, while the width of the box gives us an idea of how closely the other examples group around that median. These little lines coming out the top and bottom, those are the whiskers, and they represent the upper and lower quarters of our data, showing the range of variability. For reference, we're going to show you over here the micrograms per mil on the y-axis of the oxytetracycline. Our target dose is usually around 2 to 3 micrograms per mil for us to be seeing sensitivity of these organisms. But here's the tricky part. Higher doses above 10 microgram per mil can actually promote resistance. When we compare the two methods, the median oxytet concentration with Protectabee was a safe 4 micrograms per mil, while the conventional method was closer to 10 micrograms per mil. And that had a huge range of variability from sample to sample. In other words, bee vectoring not only hit the sweet spot for effectiveness, but it did so consistently. The takeaway, bee vectoring provides more accurate dosing, reducing the risks of under or over treatment, and ultimately it's a safer and smarter option for antibiotic delivery inside the hive. And let's also just talk about when we're often using these antibiotics. So both EFB and AFB are prominent in spring. Our organizing body, the Ontario Beekeeping Association, recommends 3 oxytet treatments 4 days apart for a prophylactic treatment of American foulbrood. And that needs to be started at least 4 weeks before the honey supers go on, because you don't want antibiotics in your honey. We actually get testing for it to make sure there are no antibiotics in the honey. But the problem is that spring is cold, wet, and honestly not the best time to open up the hive. We found that in just a few hours, powders can often clump due to moisture. In our bee vectoring experiments, we learned to add rice to the inbox so that it would act as a desiccant to dry up those powders. Traditional methods often lead to mistreatments, but with bee vectoring, you don't need to open the hive, disturb the bees, or chill the brood. And the best part, it's super fast, rain or shine. So today we're talking to veterinarians, and this is where we need your support. Now, we could just add antibiotic use with bee vectoring to the label of Tylosin, Oxytet, and some of these other antibiotics that are used for bee health. However, this would require a ton of trials with active infections, and that's actually really expensive. And antibiotics companies aren't exactly jumping at the chance to add this onto their labels because, to be honest, the amount of antibiotics that beehives require is nothing compared to large livestock. So what we're looking at now is this off-label prescription by veterinarians. This path is quicker, more achievable, and it actually just needs solid scientific backing. So now that you've got the scoop, we're ready to work together to get veterinarians on board and bring bee vectoring into the spotlight. Let's open some doors and make hive health a whole lot easier. So, exciting news, our publication is in preparation and will be out before the end of 2024. It includes all the data we've discussed today and a little extra. This is the solid scientific evidence needed for veterinarians to prescribe antibiotics through bee vectoring off label, and it'll be ready for the next beekeeping season in 2025. If you'd like a copy when it's published, just reach out and drop an email to info@bestforbees and we'll make sure you get it as soon as it's available. So let's recap why bee vectoring is such an improvement over traditional methods. First, no heavy lifting. Your beekeeper's back will thank you. It's also super fast. It literally can be done in seconds, saving you time. Spring weather, rain or shine, bee vectoring works perfectly. Plus accurate dosing. You guys saw the data. It hits the target and helps prevent antibiotic resistance by delivering just the right amount. And finally, no more scared beekeeper syndrome. They don't have to open the hive or disturb the bees to do the right treatments. In short, bee vectoring simplifies everything, making treatments more effective and less stressful. In the meantime, we sold over 5,000 Protectabees, and in 2023, we won silver in the world for beekeeping innovation at Apimondia. You might wonder why such excitement if there aren't any Varroa treatments yet. Well, the magic is actually in that red color. The red color acts as the ultimate robbing screen, protecting bees from wasps, hornets, and even other bees that might be dropping mites off inside the beekeeper's hives. Based on feedback from other beekeepers, we developed inserts that allow the protective bee to be used year-round for feeding, moving, and pest control. And now it has over 12 different uses. We've been able to use the cells of the protective bee to continue to fund our research, and now we have over 20 retail beekeeping stores carrying our product. We're excited, and so are the beekeepers. Now, all we need is the right product and veterinarians on board for prescribing antibiotics for bee vectoring. We're also open to collaboration. If you have ideas or tech that we could work with Inspensing, let's connect. Thank you for taking the time to listen today. Like a beehive, there may be a queen, but it's the workers collaborating and working hard who make the hive thrive. The success of this project has been no different. It's thanks to the teamwork of so many people. And of course, a special shout out to Dr. Peter Kevin, whose vision started us down this path. And let's not forget the sales of over 5,000 Protective Bee units have been funding our research. And we are so incredibly thankful to the beekeepers who are willing to try new innovations. Thanks again, and we're excited to keep this hive buzzing.